Hello and good evening. Welcome to this online session, online webinar today, organized by Lung Cancer Europe about treatment decisions and strategies for patients living with mutation-positive lung cancer. My name is Alfonso Aguaron, and I will be moderating today's session. Today, it is my pleasure to introduce you to our speaker, Professor Sanjay Popat, Consultant Thoracic Medical Oncologist at the Royal Marsden Hospital in London. So, Professor Popat, it's again a pleasure to have you with us, and I would like to thank you very much for your valuable and precious time today to guide us through such an important topic on how the treatment strategies are determined to treat those patients who live with a mutation-positive lung cancer. So before we get started, I would like to give our audience a brief overview on the webinar dynamics. So the session will last about an hour. And during the first part, Professor Popat will provide a comprehensive review into this topic, which will last around 40 minutes. And then in the second part, you will have the possibility to ask your questions. So to do so, you have two ways. If you would like to make your question live by using your microphone, you just have to press the raise hand icon in the panel of the application you just downloaded to join the webinar, and I'll unmute you when it is your turn so you can speak freely. Or otherwise, if you prefer to send your questions in writing, you can do so by using the Q&A of the chat window, and I'll read those questions to our speaker. So being said that, and without further ado, I wish you all a very fruitful session, and Professor Popat, I give you the floor. Thank you.
Thank you, Alfonso. Thank you, colleagues, and good morning, good afternoon, good evening, wherever you are. And it's really lovely to be able to present again a webinar for all of you on behalf of Lung Cancer Europe. I've been asked to speak about treatment strategies and sequencing for people living with mutation-positive lung cancer, and this is a very complex area with a lot of evidence and a lot of science. So let's talk through what we do and why we do it, and mention some of the controversies. So these are my disclosures. Now, first thing, I just wanted to note a few things. This presentation will be restricted to discussing the treatment of metastatic or stage 4 non-small cell lung cancer. It gets even more complex when we're talking about stage 1 to 3. Not all drugs that I talk about are available in all jurisdictions that you may be listening for, and there is considerable variability about this depending on your healthcare structure. And the drug that you may be taking may well be appropriate for your individual case. Everybody is individual. The benefits of one drug over the other may well be marginal, and the choice of drug may well be argued between oncologists. So there's often not necessarily a gold standard to potentially use. And finally, the contents of this webinar just reflect my own personal views only. So non-small cell lung cancer, what is it? Well, it's a diverse group of different diseases driven by different molecular alterations. If we take a group of patients with non-small cell lung cancer stage 4 on the left-hand side, in fact, they can be split into different categories of non-small cell lung cancer depending on their driver genetic alteration in their tumor. This is the first genetic alteration that occurred in the original cell that became cancerous, which is then present in the daughter cell when that cell divided and become 2 cells, and 2 cells became 4 and 4 and then 8. That genetic event is still present in each and every cell. And that genetic event is driving that cancer to grow and divide. Fortunately, these days, since the late 2000s, we have a class of drug which can often switch off the function of that genetic event, and these are called tyrosine kinase inhibitors or sometimes kinase inhibitors, and these are broadly tablets. And that can result in often dramatic shrinkage because that is the driver genetic abnormality which is causing the problem, which is then switched off, and that genetic abnormality is present in many cells. In the top right-hand panel is a CT scan of a patient with ALK positive, ALK positive lung cancer. You can see this is in fact a lung, a slide with lungs. These should be black areas, but in fact full of white areas, which is the tumor. She was shown to have a ALK genetic alteration and started on a kinase inhibitor, and you can see that the disease melted away beautifully in a short period of time. And now this is entirely standard. And in fact, if we look at where we are in advanced non-small cell lung cancers, there are at least 8 different genetic alterations, at least 8 genes for which this genetic alteration should be looked for to enable us to use either FDA or EMA approved treatments. And that is looking at the genes that are EGFR, ALK, ROS1, BRAF, NTRK, MET, RET, and KRAS. All of these we have drugs that are potentially useful. Some would argue that understanding the HER2 status is also important, with encouraging activity with drugs in that sector, but yet none of these drugs are currently approved for lung cancer by the FDA or in Europe. And alongside all of this, the tumor should also be tested for PD-L1 status to help the decision making for what we do. So of course, we can take a patient's tumor, we can analyze it, we can find that they have a genetic alteration in ALK or EGFR, we can give them a tablet which shrinks it down, and everybody's happy. The patient's happy, the patient's family is happy, the oncologist is happy, and their team are happy. The problem is that often what happens is then the tumors are clever, and they bypass the activity of that drug. And after a period of time, maybe it's a handful of months, maybe it's a handful of years, the cancer starts growing again. And that is called acquired resistance. And then we need to work out what the next best steps are for that patient. So there are many causes for acquired resistance that are what we call on and off target acquired resistance mechanisms. These mechanisms can be pharmacological, to do with a drug. Maybe the patient's not taking the drug. Maybe the side effects are so difficult they just don't want to take the drug anymore. Maybe the absorption of the drug or the metabolism has changed. Are they on other drugs which are increasing the metabolism of the actual anti-cancer drug? Are they taking drugs or food which interact with the absorption of that particular drug? Or in the right-hand panel, perhaps the most common, the drug just doesn't get into the brain at decent enough a concentration, because the brain has lots of pumps around it which pump out the drug and make sure it doesn't accumulate into a big enough concentration, because the brain likes to be kept pristine, away from the toxins of the rest of the body. But there are also biological reasons for acquired resistance, which we can see, which may be because the drug target itself, such as ALK or EGFR, has become abnormal. Is there a resistance mutation that has occurred within the target to cause the resistance? Is there more copies of the same gene to cause resistance, or other genes have become activated in the same cancer to cause signaling of the abnormal signal through to the nucleus? These are called on and off target resistance mechanisms. There are other things that can happen. Maybe the cancer can change fundamentally, to change from being a non-small cell lung cancer to a small cell lung cancer. So it's important, perhaps, but not always, in patients with targeted therapies to understand why the resistance occurs. And perhaps this then opens up a new abnormality which we might be able to exploit to have another drug used in that scenario. Now, there are areas where this is definitely a helpful thing to do, such as looking for the EGFR T790M mutation. If perhaps you're in a first or second generation EGFR inhibitor like Latinib, a first generation inhibitor, if you get acquired resistance, maybe T790M is the altered drug target, and that opens up a utility to use osimertinib. And in the ALK setting, we're not sure whether this is helpful, such as resistance on alectinib may be useful to deriving the next drug, or perhaps if you're on osimertinib with resistance or growth, that might be useful for determining the next treatment, but not necessarily mandatory. Nice to know, but not need to know. At least people argue about that. Now, there are 5 basic principles of targeted drug treatment for genetically altered non-small cell lung cancer that you should know. In general, the first principle is that we use the most effective drug first. And when that stops working, we use the second most effective drug second, and so forth. However, many things can happen to prevent patients getting more treatment. Patients can unexpectedly deteriorate. They can develop unusual complications or brain metastases which prevent more treatment. And that's why we use the most effective drug first. We tend not to keep the most effective drug up our sleeve. Second, patients with genetic alteration-driven non-small cell lung cancer usually have a subtype called adenocarcinoma, and these patients have either moderate or high risk of brain metastases either at baseline or at some point in their lifetime. And not all targeted treatments cross into the brain to treat the brain metastases, so we need to think about how we can manage this problem. Third of all, failure of the first targeted drug because of acquired resistance after a period of time after the first drug, we need to understand if the biology has changed. Is there a new genetic alteration? Can this new genetic alteration be drugged? Or should the same target be hit harder? Is there a more effective drug that has now come out since the patient was on the first drug, which means we can hit it harder and treat again effectively? Are we treating the brain only, or are we treating the body, or both? Perhaps the disease is stable in the body but progressing in the brain, which needs a slightly different approach, or stable in the brain but not the body. It needs a different approach. And is there a targeted treatment that's the next best option to use, or is chemotherapy the next best option? Point number 4. Chemotherapy can actually be very effective, and in some patients with genetically altered disease, it's actually more effective than in patients without genetically altered disease. And in general, this is the standard that we use when we don't have a targeted therapy option. And finally, the principle is that immunotherapy has little or no activity for most genetically altered non-small cell lung cancer in general, although there are some exceptions. So how do we know all of this? Well, most of this has become from our scientific understanding of EGFR mutant non-small cell lung cancer, which was the first major druggable type of genetically altered non-small cell lung cancer that we identified. And then as other genetic alterations have been identified and understood, and as new drugs have been developed, we've understood these further principles. So let's take our sequence, and I've taken the NICE guidelines, which give us actually very good, I think, guidelines for how we should cost effectively sequence the drugs that we use. So let's look at the example of EGFR mutant non-small cell lung cancer. NICE suggests we can use afatinib, dacomitinib, erlotinib, or gefitinib and first, and then if T790M positive, move to osimertinib. An alternative is to start off with osimertinib first of all. And then when that fails, the next step at the end of the dotted red dots is in fact chemotherapy. So when we're talking about EGFR mutant lung cancer, we have to actually recognize there are many different mutations within EGFR, and our data and understanding and how these behave are slightly different. We generally talk about common EGFR mutant non-small cell lung cancer, so something that's called deletion 19, exon 19 deletion, or L858R. And if you've been on a first or second generation EGFR kinase inhibitor, at time at which that stops working, not all patients, but several patients can develop the T790M mutation. So L858R can also acquire T790M, so called L858R plus T790M. There's the other group called EGFR exon 20 insertions, for which I'm not going to talk about further, and there are new drugs being developed, licensed by the FDA, not yet by EMA. We have point 3, the major uncommon EGFR mutants, lung cancer. So these are genetic alterations such as G719X or S768I, which are not common. Then you have much rarer EGFR uncommon mutations such as L774X. And then you can have mixtures of all of these. These are called compound mutations, EGFR exon 19 deletion plus G719X, or for example, a rare plus rare, G719X plus L861Q. So how do we treat this disease? Well, I'm going to focus on common EGFR mutant lung cancer. Originally, we knew that TKIs, EGFR TKIs, are much more effective over chemotherapy. And there are 8 phase 3 trials, if not 9 or more, which have confirmed that using either gefitinib or erlotinib or afatinib over chemotherapy is superior. And this table shows that something called median progression-free survival, or the time from starting treatment to relapsing, is much longer for the TKI than it is for chemotherapy. And the proportion of patients having tumor shrinkage, that's the response rate, is much higher for TKIs than with chemotherapy. But with these drugs, we were never able to prove that survival was longer. So we'd like to give it on the first line setting because it makes rapid improvement in patients, but it didn't prolong the survival of patients that started with chemotherapy and then may have had an EGFR TKI thereafter. At point of progression, let's say you're on gefitinib or erlotinib or afatinib, we were then in general giving chemotherapy. And some were arguing, well, why don't you just carry on the EGFR inhibitor and add in the chemotherapy? And that became very popular, particularly in parts of North America. And this was formally examined in a trial called the IMPRESS trial, where half the patients were allocated just chemotherapy alone, and half the patients were allocated continuing with gefitinib. In the left-hand plot, we can see that the outcomes for progression-free survival, or the time to progression, were the same for whether you just had chemo or chemo plus gefitinib. So carry on the gefitinib didn't really give you any additional benefit. However, on the right-hand panel, what it showed is that this was setting up patients for inferior survival. Patients were actually surviving shorter when given the combination of gefitinib with chemotherapy than just chemotherapy alone. And that prompted a heavy move away from the TKI plus chemotherapy to just chemotherapy alone. So this is in the acquired resistance setting. Then we started to understand a lot more about the biology of EGFR resistant cancers. And when we biopsied these patients that had progressed on gefitinib, erlotinib, or afatinib, or even decomitinib, we identified that around 40 to 50% of patients had acquired a new mutation in EGFR called T790M. And in the remaining group of patients, there were either no obvious resistance mechanism. It's important to know that. Sometimes we just don't know what the resistance mechanism is. Or there were resistance mechanisms that are not druggable, or the patient's disease had changed to become very aggressive and changed to become a small cell lung cancer. Now, it's important to know if T790M is there or not, because at the same time, AstraZeneca were developing a drug called AZD9291, which is now called Osimertinib. And this drug was specifically designed to switch off and inhibit T790M EGFR mutant lung cancer. And in a phase 1 trial, when we took patients that were either T790M positive or negative, the T790M positive patients had much better progression-free survival time from starting drug to relapse compared to patients that were T790M negative. That was then formally evaluated in a trial called AURA3, which took patients that had relapsed after first-line gefitinib, erlotinib, afatinib, or dacomitinib, and were then biopsied and shown to be T790M positive. They were randomly allocated to either chemotherapy or osimertinib. And the progression-free survival was much longer with osimertinib than chemotherapy, with a median of 10 months compared to 4 months, making it then the standard of care, which is why if you're on one of those earlier drugs and you relapse, it's so important to know whether you have T790M or not, because we would want to use osimertinib in that setting. And then that begged the question, well, if osimertinib is so good, why don't we just use it up front? And so we then did a trial called FLORA, where we took patients which had newly diagnosed common EGFR mutant lung cancer, DEL19 or L858R, and randomized them to gefitinib or erlotinib, or the new drug, osimertinib. And you can see in the plot that the osimertinib patients had a much better progression-free survival compared to standard TKI. In fact, their disease was under control for a median of 18.9 months compared to 10.2 months for the standard erlotinib or gefitinib. And that then posed the conundrum on the right-hand side. Do you then start off with the first-generation TKI for 10 months, work out whether it's going to be T790M positive or not at time of relapse, and if T790M positive, give osimertinib for another median of 10 months, whereas patients that are negative get chemotherapy where they have a median of control for 5 months, or a second-generation EGFR inhibitor which is slightly more active, median of 14 to 16 months, followed by osimertinib for 10 months if they're T790M positive. Or do you avoid all the T790M testing and the toxicities of second-generation inhibitors and give them osimertinib up front for a median of 19 months? And osimertinib is also extremely good because its side effect profile is much better than the other drugs, and it has very good brain penetration. It's actually designed to go into the brain. That became a point of contention since 2019. This data were presented. 2018 that these data were presented until 2019, when the overall survival of these patients was presented, because these had then been followed up, and it was clear that if you started off with osimertinib compared to the standard EGFR inhibitor, patients were living longer. So osimertinib then became the standard as frontline setting if you were newly diagnosed with EGFR mutant non-small cell lung cancer. However, some have argued that in this trial, if you look at patients that were started on gefitinib or erlotinib and then developed T790M, they may have actually had a similar survival, but the patients that crossed over to receive osimertinib in the control arm weren't that many, and therefore you're not comparing like with like. You're not comparing the sequence with osimertinib, but the osimertinib believers, and I'm in that group, will say, well, look, osimertinib has much better brain penetration, and you cannot predict up front whether the patient is going to develop T790M. So you always use your most effective drug up front, which is why osimertinib's used up front in general these days. So what do you do when osimertinib stops working? So we use osimertinib in two settings. We use it up front as the first drug, and we use it as the second drug in patients that have had one of the earlier drugs. So in the left-hand panel is the selection of genetic alterations which occurs when we've rebiopsied a group of these patients that had a first-generation or second-generation drug, then developed T790M, then went on osimertinib. This is what we're seeing in the left-hand pie chart. And what we can see is that the genetic resistance mechanisms to osimertinib are multiple. There's not one dominant genetic alteration. Several studies have said that MET amplification can occur up to 50% of patients, and that is something that is now being evaluated further. There are other difficult genetic alterations in EGFR that can happen, C797X and others, and these are not really druggable by other conventional EGFR inhibitors. But you can see that many different genetic alterations can happen, and in fact, in some patients, in the green panel here, 4 to 15% of patients, the disease can change to become more aggressive and become small cell lung cancer. We are now looking at trials to switch off these novel EGFR genetic mutations, and also combinations with MET inhibitors to see whether this improves things. And in patients in the right-hand panel, if they have been started on first-line osimertinib, we see a similar spectrum. We don't see T790M. We see sometimes that the original EGFR is lost, and we see MET amplifications coming through as well. So currently the standard for patients at this point is to switch to chemotherapy because we have no approved druggable combinations, but we always try to see if we can enroll patients into clinical trials such as MET inhibitors to see whether we can use a novel targeted therapy before chemotherapy. Should these patients receive immunotherapy? Well, the answer, I think, is no, because if we take a group of patients, and this was a phase 2 trial of a handful of patients with EGFR mutant lung cancer, a trial done in California, every single patient did not benefit from immunotherapy, and the response rate to immunotherapy was 0%. So in the setting of upfront disease, all patients should get EGFR inhibitor, and when relapsing without any more targeted therapeutics, they should get chemotherapy because immunotherapy by itself has no proven value. And this underpins the NICE treatment recommendations for the sequence. Let's look at the ALK field. Things are much more complex here. We know that there are 4 different ALK inhibitors that one can go on at time of diagnosis according to the data. And Consultan is not yet approved for use globally. And most patients start alectinib, although there's very good data to start with brigatinib, and Latinib is approved in the US and by EMA in the frontline setting, so you might want to consider that as well. But all drugs are highly, highly effective with a median progression-free survival time from starting to relapsing of between 2 and 3 years, if not longer, compared to the standard, which had been the first generation EGFR inhibitor, ALK inhibitor, CRSOtinib. But what do you do when things relapse? Well, we know that each of these drugs are very, very good, and it doesn't really matter which of these drugs you're on. Each oncologist will have a slightly different preference, and they will discuss that with our patients and make shared decision-making. And not all of these drugs are available in all jurisdictions. Each of these drugs has slightly different brain penetration, and that may be important in the decision-making. Importantly, each of these drugs also has different schedules. Alectinib is 8 pills a day, brigatinib is 1 pill a day, Latinib is 1 pill a day. They all have a different side effect profile. They have different rate of side effects needing dose reductions. The side effect profile are very different between these 2 drugs. Alectinib tending to cause constipation, muscle aches, abnormal liver function, brigatinib causing biochemical abnormalities, causing high blood pressure, can cause a cough, can cause rare breathing problems at time of starting treatment. Latinib has problems with its lipids, cholesterol, triglycerides, fluid retention, weight gain, memory, thinking, neurocognitive problems. So that's an individual discussion with your oncologist as to what to do. But when these patients relapse, if we rebiopsy their disease, if they've been on alectinib in the left-hand setting, we can see that the predominant resistance mechanism is something called G1202R. And if patients have been on multiple ALK inhibitors and then finish with Latinib, they can have multiple ALK mutations, but not every patient gets a ALK mutation coming through as a resistance mechanism. So does it matter whether patients have these resistance mechanisms or not? Well, we know that these different drugs can have different likelihood of benefit. In the green is highly active according to a different mutation, yellow is moderately active, and red is inactive. But this is a cell line model. This is not human beings, and this doesn't take account of brain penetration. And none of the trials we have really looked at whether patients are deriving benefit according to their mutation status at time of enrollment. So therefore, whether treatment directed by ALK genotype is clinically useful or not remains a matter of oncological debate and is the focus of an ongoing US trial. So at the moment, we tend not to treat according to mutation status. It's interesting to know because it may alter your decision-making, but it's not mandatory to know. We tend to use Latinib if you started on non-Latinib drug starting up front. And we know with lorlatinib, if you have an acquired resistance mutation, then you have a much better progression-free survival of around 11 months going into it, particularly if you have the alectinib-resistant G1202R mutation. Whereas if you're relapsing without a mutation, then the survival benefit, progression-free survival, the duration of treatment with lorlatinib is much lower at 5.4 months. And after that, what do you do? Well, you may be able to salvage more TKI if you have a different mutation, but otherwise, we're generally using chemotherapy in that scenario. What about patients with ROS1 positive disease? So here we don't really have a good sequence. Patients are started either on crizotinib or entrectinib, although there is some data for salvage with lorlatinib, but it's not approved by any jurisdiction for ROS1 positive disease. So what is our data to support this decision making? Well, we have two trials, or several trials looking at crizotinib, which is a very good ROS1 inhibitor. This is data from the Profile 10001 data, which is updated on the right-hand panel, and you can see that nearly 70% of patients are responding or tumor shrinkage. Each line here on the waterfall plot representing the magnitude of shrinkage of an individual patient. And that shrinkage is durable with a median progression-free survival of anywhere between 14 to 20 months, depending on the trial that you're looking at. So crizotinib is a very good drug for ROS1 positive disease. However, we know that crizotinib does not really penetrate into the brain that well, and often these patients then relapse with intracranial progression or disease growing in the brain. Or if patients have brain involvement at time of progression, often it has limited activity in the brain and patients can relapse in the brain thereafter. Another drug which is often used is entrectinib. It's much more potent at ROS1 than crizotinib, and it also has good brain penetration. It's also a very good NTRK inhibitor. The data to support it, because ROS1 is very rare, it occurs in about 1% of all our lung cancers, there's very little trial data to base our decision making on. The registration study of entrectinib was based on 32 ROS1 fusion positive patients, with each patient getting very good shrinkage, which translated to a median progression-free survival not of 10 months or 15 months, as we're seeing with crizotinib, but around 30 months in this highly selected group of patients in the entrectinib Star Trek 1, Star Trek True, and ALCA trials. And this is the data that we use to use it up front. But what about when patients relapse? We have good data on the molecular status of when they relapse on crizotinib. In half the patients, there isn't a change to the genetic target. And half the patients, they acquire another resistance mutation called G2032R. And if you have one of these mutations, there is data to use lorlatinib thereafter. Lorlatinib has activity in ROS1 positive non-small cell lung cancer with response rates of around 26% in the body, but in 52% in the brain. And the median progression-free survival is around 8.5 months in patients using it after another ROS1 inhibitor. Unfortunately, Pfizer have not taken this forward, and it's not approved for use in ROS1 positive lung cancer, so it's hard to access this drug in many healthcare jurisdictions. What about BRAF V600E? This is a very rare and aggressive type of lung cancer where we use BRAF-MET combination inhibitor treatments. Dabrafenib and trametinib are two kinase inhibitors. They're not tyrosine kinase inhibitors. They're kinase inhibitors which switch off BRAF and downstream signaling through MET. When you give these two drugs together in the second-line setting, 64% of patients are getting durable shrinkage which is lasting around 11 months or so. And if you give it in the first-line setting, we're seeing the same thing. Around 67% of patients with very good shrinkage, durable, lasting around 11 months or so. We don't really have an approved drug to use in the relapse setting, and these patients thereafter are generally treated with chemotherapy with or without immunotherapy. What about another rare subset, Repfusion positive non-small cell lung cancer? Very rare, around 1% of all what we see in non-small cell lung cancer, mainly in never smokers. We have 2 approved drugs in Europe and in the US, selpercatinib and pralsetinib. Both drugs are used in any line of treatment in the US, and in Europe, pralsetinib any line and selpercatinib in the relapse setting. These are highly effective RET inhibitors, and with selpercatinib in the platinum pre-treated population, the relapse setting, 64% of patients responding with a very good, durable 19 months progression-free survival duration of treatment from the Libretto 001 trial. And in the treatment-naive setting, patients presenting up front, we don't have that much data, but 85% of patients showing very good shrinkage, and the progression-free survival hasn't even been reached for those patients in the trial. We have very similar data with pralsetinib, a very similar drug with slightly different side effect profile from the ARROW trial in platinum pre-treated patients in the relapse setting, showing response rates around 61% and 73% in the treatment-naive setting. What about MET exon 14 skipping mutant non-small cell lung cancer? Patients say to me, well, what is this MET 14 skipping? It just means that in the genome we have areas where the genes translate into a protein. They're called the exons, and the junk in between called the introns. And this genetic abnormality causes the exon 14 to be spliced out, cut out, and then skipped out. That's what it just means. And there are 2 drugs which are approved for use, capmatinib and tepotinib. They are approved for use by the FDA in any setting, and not approved for use at the moment by EMA. Capmatinib, the data to support it are from the Geometry Mono 1 trial, in which patients with relapse disease had response rates of 40% with a progression-free survival of about 5 months. So not as active as we've seen in some of the previous TKIs. And in patients in the first-line setting, a higher response rate, around 70% of patients on it with a progression-free survival of around 10 months or so. And in the VISION trial for tepotinib, we're getting very similar response rates and progression-free survival. We don't have targeted therapies suitable for when these patients relapse, but we are starting to understand the genetic mechanisms of resistance better. And in general, these patients are treated with chemotherapy with or without immunotherapy thereafter moving forward. So with that, I'd like to thank you. I hope you weren't too bored and found that of some interest. And back to our host, and I'm very happy to take any questions as they arise.
Thank you so much, Professor Popat. As always, it's been such a clear and comprehensive presentation. We have received some thanking messages talking about the clarity on explaining this topic. Even though it's complex, I think you have done it in a very easy to understand way. So we have already received a few questions that I'm going to pass over to you. I just would like to remind our audience that if you have any questions, you can send them in writing by using the Q&A or the chat window, or if you prefer, you can press the raise hand icon and I will unmute your microphone so you can make your question live. So the first question is about one of the drugs that you have mentioned, which is Sorotacib. And the person is asking, when do you expect Sorotacib to be approved in Europe for KRAS patients?
Yeah, that's a really good question. So Sorotacib is a what's called a KRAS G12C direct inhibitor. So this is not a tyrosine kinase inhibitor. It works in a slightly different way, but it switches off not just a KRAS gene, but a very specific KRAS mutation called G12C. And this is one of the commonest mutations we see in KRAS. Lung cancer, about 30% of lung cancers have KRAS mutations. There are different types of KRAS mutations, but one of the commoner ones is KRAS G12C in lung cancer. There is data on a drug called Sorotacib, which is produced by Amgen, which has got accelerated approval in the FDA. The UK MHRA, which is now outside EMA as the regulatory authority, have already approved it, so we can access it in the UK. But in the rest of Europe, we are still awaiting European approval. I hope it's imminent. I hope it's within the next few months, because I know they are looking at it, and I hope that we do get approval for use in Europe. I remain optimistic.
Thank you so much. I think maybe I'm not very familiar with this, but I think this question is linked to that, because one of the person is asking, is there any update on the AMG 510 trial results? So I think this is the trial that you were mentioning, right?
Yeah, that's the AMG 510 is, yeah, that's exactly the same name as Sotatercept. AMG 510 was Amgen's code number for the chemical, which has now called Sotatercept. And you know, it showed what we have had presented with Sotatercept is data to show that in patients that have had previous treatment, so previous treatment with immunotherapy or chemo or immunotherapy in combination with chemotherapy, it can cause shrinkage in a proportion of patients, and a progression-free survival is not quite as good as we usually see with kinase inhibitors, but that's because this drug works in a very different way. We don't know, because we have no randomized data to compare it to the drug that we would normally use in this setting called docetaxel, how much better it is than docetaxel, but we think that it's going to be much more effective than docetaxel chemotherapy, which is why we're all excited to try and use it. And the randomized trial data should be available next year.
Thank you so much, Professor Popat. So next question from a person who's asking, even your presentation was based on a stage 4 non-small cell lung cancer, I would like to ask, are there any biomarkers present in a small cell lung cancer? And he also asks, may we see targeted therapies for this particular group of patients in the future?
It's a very good question. I mean, small cell lung cancer is a very difficult disease, and our understanding of small cell lung cancer has really been scuppered quite a lot by the fact that it's very difficult to take tissue from patients. The disease tends to be very central in the airways, so actually getting samples of tissue is really challenging. And that's really limited a lot of our understanding of the biology. Nevertheless, there are some really good advances coming through in the biological understanding of small cell lung cancer. The one big change that has happened with small cell lung cancer over the past 2 years has been the introduction of immunotherapy. And the addition of immunotherapy to chemotherapy for metastatic or stage 4 small cell lung cancer is now standard for patients that are fit and suitable to have this. They should all be getting chemotherapy together with immunotherapy. We don't really see the same genetic alterations with small cell lung cancer as with non-small cell lung cancer. So in non-small cell lung cancer, the genetic alterations switch on the cells to cause the cancer. And so therefore, if you give a tablet which switches off the genes, that causes the lung cancer to shrink down. That's what happens in non-small cell lung cancer. In small cell lung cancer, the genetic alterations don't switch it on, they take off the brakes. They take off the brakes so that other processes then take over to cause the cancer to grow. And so we can't really give these drugs to switch off the genes, because the genes are already switched off. They are then taking the brakes off these other processes. So we need to think differently about small cell lung cancer to non-small cell lung cancer. There are some targeted treatments which are coming through. There are some data looking at what are called PARP inhibitors, which are being developed. There are new types of chemotherapy being developed, and there are new targeted treatments such as lubrinectedin, which is being developed, which may have a role in the future in small cell lung cancer. So I still think the future for small cell lung cancer is still very exciting. There are new drugs and combinations being developed. Watch this space. because I suspect over the next 2 years, how we treat small cell lung cancer will be very different to how we treat it now.
Hopefully we'll get that soon, because these patients are in a very urgent need. So hopefully we'll see a change on that. So next question is quite interesting, and it's coming from a person, but this is something that we sometimes are receiving here in Lung Cancer Europe, and I would like to know your opinion, Professor. So what's your view on the use of generic TKIs manufactured outside of Europe, especially in Asia, to grant access to certain drugs which can be difficult to access in certain countries? And this person is writing from Croatia.
Yeah, I mean, I think, I share the pain that our patients have because the cost of drugs are prohibitive. We have this as a major problem in oncology, is the cost of drugs are prohibitive, the cost of development of drugs are prohibitive. And because the costs are prohibitive, we have such rigorous application for when we use drugs and when we can't use drugs. The challenge, I don't mind using a branded drug, but the challenge I have with Me Too generic drugs is if it's a established generic off-patent drug, because some drugs are now off-patent. Gefitinib is now off-patent in certainly in the UK. Then I have no problem with using an off-patent drug as long as it is manufactured to a regulatory standard. The problem that we have is that many of these drugs that are coming through, which are generic, are not manufactured in a regulatory controlled manner. So is the patient actually taking what it says on the pack? That's the real key issue, because we don't want patients to get side effects or undertreatment if what they're taking on the box isn't what's inside. And so my problem that I have with a lot of some of these generics that come from elsewhere is we just don't know what it is. We don't know whether what it says on the pack is actually what's in the tablet. So if you're using generics, if that's an appropriate generic, manufactured in a recognized, regulated, FDA approved, or the country's approved system, then I don't have a problem. It's when it's unapproved, then we don't really know what we're being treated with.
And may I ask you, Professor, is there a way for patients to really know this? I mean, whether even because I understand this drug is going to be prescribed by a doctor, but is there any way that they can really make sure that what they are getting, especially if it's a kind of generic drug, really contains what it said in the package, that it goes under certain quality controls or so on?
Yeah. I mean, the branded drugs are branded. So, if you buy a Latinib from Roche or Chugai, it will have their marking on it. If it's manufactured in Europe, it will have a CE mark on it. US approved drugs will have an FDA label package insert in it. At the end of the day, it sort of depends on your relationship with the pharmacist or the oncologist, who will know, your local oncologist will know where that drug is coming from, or whether that particular brand is manufactured in a regulatory manner. All countries keep a list of regulatory approved products. So go to the website of the drug regulator, which will keep a list of the name of the drug, and find out whether that brand is an approved brand or not.
Great, thanks. Thank you so much for that clarification. So we still have a few questions left in here. Next one, it's something completely different. So this person is asking, in which cases a patient with an oncogene-driven mutation might be eligible to receive radiotherapy instead or complementary to a targeted therapy?
So yeah, thanks. So we use radiotherapy in many different ways. We use radiotherapy with curative intent in stage 1 lung cancer, stage 3 lung cancer as well. And in stage 4 lung cancer, we generally use it in what's called palliative intent to help the symptoms of cancer. So if patients have pain, we give low-dose radiation to a bone or an area to help take off the pressure, to help with pain. If they have a tumor that's causing compression of the airways or bleeding, we can give low-dose radiation or palliative radiotherapy to help there. What is really interesting now is the advent of what's called SBRT, or stereotactic body radiotherapy, which is very focused, high-energy, radical radiotherapy to a very tight area. And the key issue is, where should we be using this, and does this make a difference to patients' lifespan? Does it change the natural history of what we're doing? So one area where we're starting to look at this is, if you have widespread metastatic disease with oncogene addiction and give a TKI, let's say EGFR, everything melts down beautifully. You're left with very small disease. And we see relapse. We generally don't see widespread massive relapse. We see either small volume relapse occurring in a few sites, or we see this unusual pattern called oligoprogression. Oligo is Greek for few sites progressing. So maybe you'll have everything brought under control, but one or two cancerous deposits in the lungs will start growing a little bit. Now, the key question then is, most oncologists would not change drugs at that point. You know, they wouldn't say, well, look, this is meaningful progression. Just carry on and we'll keep an eye on it. And then when we need to, we might be able to biopsy it, understand the biology, switch to the next drug, or if there isn't another next drug, use chemo then. The key issue is, if you have oligoprogression, should you be using stereotactic radiotherapy to blast those two areas with highly focused radiotherapy to then allow you to continue on the drug for much longer than you would have done otherwise? Now, using stereotactic radiotherapy has become very popular in the US and many parts of the world, but we don't really know what it's bringing to the equation. How long extra are we getting on the TKI for this radiotherapy on average, or are patients just undergoing and paying for this radiotherapy for no additional benefit? So in Canada and in the UK and in Europe, we're doing a trial. In Canada, the trial's called STOP, and in the UK and Europe, the trial's called HALT, where we're taking patients with this oligoprogression and randomly allocating them to either to continue the TKI, keeping a careful eye on them, or give them the SBRT to the radiotherapy to see whether it helps. I hope that answers the question.
Yeah, thanks. I think it was very clear. I would also like to add to this person that if you would like to know a little bit more about how this SBRT or stereotactic body radiotherapy works, in our YouTube channel and in our website, you can find the recording of a webinar that we held a few months ago about the use of radiotherapy in lung cancer, and all these technique is very well explained also in there. So it may also help to complement your very good answer. So we still a few questions left. Next one is, would that be possible also in Europe to use the combination of lorlatinib plus chemo after lorlatinib, like in the US for ALK patients?
Oh, very hot question. Is probably the commonest question I get asked, actually. So it's not straightforward to do, because the combination of chemo plus lorlatinib is not a licensed combination. It's not an approved combination. We don't know what the right dose of lorlatinib to combine with chemotherapy is. Now, in the US, they will say, well, you're on lorlatinib and your cancer's relapsed. Let's just add in the chemotherapy and carry on the lorlatinib. I'll remind your listeners of the IMPRESS trial data that I showed you, where people were doing that in EGFR about 10 years ago, and the trial showed that actually the combination had an inferior survival rather than just using the chemotherapy alone. So carpe diem, seize the opportunity. It might be something worth considering, but buyer beware, because you don't know what it's bringing to the equation. Outside the US, in Europe, certainly we have a very regulated system where we can't just take things off the shelf and add them together. We have to do things according to the regulators, and that, because at the end of the day, that's what approved funders will use. And because we need more data, I think this is a call out to my US colleagues. If you've done this, please publish your data, because we really want to know what this is bringing to our patients, so we have some scientific data rather than just anecdotes from our patients.
this need of evidence, I think it's quite clear. Indeed, there is a question which is, in a sense, a little bit linked with this, and I think from the patient perspective, it really makes sense. So it's a person who's asking, I can see there are lots of options in US compared to Europe, and he's asking, are there any ways to prescribe these drugs until these are included in national healthcare baskets?
The short answer is it's not easy because the US is a much less regulated healthcare structure than outside the US. Particularly, Europe is very regulated because, for example, in the UK, the government is paying for drugs, not the individual. And actually, the culture of governance is much stronger in Europe than it is in the US. We have a much stronger reliance on evidence-based medicine. If a combination of drugs is looking very, very active, sometimes the manufacturer may make it available before a license, but it's usually when there's pretty good culture of evidence body of evidence to combine the drugs together. So I think that's very much an individual discussion to have with your oncologist about your personal individual circumstance and the combination that you're looking at. But the broad answer is it's not straightforward to do that in Europe, mainly because of our culture of governance, our culture of wanting good quality evidence to protect patients from quackery and preventing crazy things from happening, and wanting to have good quality evidence to support the decision making that we give to our patients.
Thank you so much. 2 questions left, so very quick to make it on time. First one is, in which cases anti-PD-1 or PD-L1 drugs are recommended for patients with a certain mutation, if any?
So in general, patients with most mutations have no benefit from PD-1 or PD-L1 monotherapy. Some patients with MET14s may derive a benefit, but not huge. Generally, after targeted therapies, chemotherapy is best. I tend to reserve checkpoint inhibitor treatment, either never at all, such as ALKs, they never benefit at all, apart from 1 or 2 rare individuals, or use it as last line of trying.
Great. And last question, coming from an advocate that I know very well and who's strongly advocating for this. So you have mentioned quite rare and uncommon mutations. So we in the community are a little bit concerned about the difficulties in certain hospitals or countries to get an accurate test due to various aspects such as access to panel testing or innovative diagnostic techniques such as NGS. So would there be a recommendation for patients so they can be sure the diagnosis is precise and accurate?
Yeah, I think in Europe we need to genotype all non-small cell lung cancer adenocarcinomas, not necessarily squamous carcinomas, but adenocarcinomas at the 8 different molecular genotypes that I said. And that really should be standard. I'll tell you now, it's not standard everywhere. Certainly even in the UK, it's an aspiration and not a standard. Many laboratories are able to do this. Many laboratories are not able to do this. And part of the problem is sometimes there just isn't enough tissue on the diagnosis specimen to be able to do such testing. I think this is a bigger issue than just individual patients, because we are really keen to run out NGS panel testing for everybody, but it's going to take a while till we get this out there for everybody.
Great. So that was the last question, and we managed to make it on time. So thank you so much. Before closing, Professor Popat, as always, it's been a pleasure to have you with us again, and I would really like to thank you on behalf of Lung Cancer Europe for such an outstanding presentation. We have received many thanking messages in the meantime, so I would like to pass over to you. And so I'm I'm pretty sure that our attendees have found this very interesting. So before closing, I would also like to thank you all for attending this webinar, and would like to remind you also that the full content of this session has been recorded and will be soon uploaded to our YouTube channels. Remember, we're making a strong effort to have these available in different languages, so we will also be including subtitles in principle in French, German, Italian, and Spanish, so we can spread the knowledge to the European community, and hopefully in the future we can include more languages to these. So you will receive an email soon with the link, so you can watch it as many times as you wish. I would also like to take the chance to encourage you to follow our activities in our website, which is www.luncancereuro.eu, as well as to follow us on Facebook and Twitter, and also just have a look at our member section to be in touch with your local patient group in case you haven't done this yet. So with that being said, I would like to wish you all a very fruitful evening, and I really look forward to meeting you also in our upcoming webinar. And thank you for attending, and thank you again, Professor Popat. It's been a pleasure to spend this evening with you. So goodbye.
Goodbye.